Hello, this is Mrs. Murdock, dressed as a turkey because it is pajama day and it's all I had. So I'm going to take this off because it's really hot. Okay. We're going to be talking about aerobic respiration today. Now you guys already have a foldable where you filled in some things about the mitochondria. What I'm going to do is I'm going to draw my own rendition of this process. And I'm going to start with the mitochondria, and as you know, the mitochondria has two membranes to it. It has an outer membrane and an inner membrane. So this is the chondrion singular, right? And a mitochondria would be in a eukaryotic cell's cytoplasm. You guys know that. So out here is the cytoplasm of a eukaryotic cell, right? Because bacteria would not have these. These are membrane-bound organelles. And there is an inner and outer membrane here. So out here, there's the outer membrane. And then there's an inner membrane. Inner membrane. And these two membranes create two very important compartments or spaces within the organelle. The innermost compartment is called the matrix. The outermost compartment, where a lot of very important things happen, is called the intermembrane space. So intermembrane space is out here. And we're going to start from the very beginning of what you learned about when we started talking about anaerobic respiration. You know that the first step, whether it's anaerobic or aerobic, is glycolysis. So every cell does this. Even the bacteria can do this, right? But not every cell can go on further into the mitochondria, right? Glycolysis is where you take glucose and you break it down and you break it down until you get two pyruvates or pyruvic acids, two pyruvates. Remember, we're starting with 6 carbons up here, and then we wind up with 3 carbons down here. So first law of thermodynamics is agreed with here. And then you need these 2 pyruvates because that is what the mitochondria is going to need to do its next steps, right? So the products of glycolysis are going to go in and then continue in the aerobic pathways. But before I do that, let me remind you that glucose gives us some products that we need to remember. So glucose gives us a net of 2 ATP. Don't forget that. Those are really important to the bacteria because that's all they get. And it also gives us 2 reduced electron carriers that we have learned about. So I'm going to use a little bit of color here. I'm going to keep the ATPs red because we like to kind of count them up to see how they're doing. And I'm going to keep the NADHs blue because they are also very important in helping to make ATP. So we're going to keep track of those as well. Okay, so now we're going to go into the mitochondria. And what happens with those pyruvates, remember there's 2 of them, and they have moved in to the inner matrix. Remember there's 3 carbons each here, right? And something happens called the link reaction. The link reaction is often not really discussed a whole lot. Certainly you didn't hear about it in 9th grade. And sometimes, I'm not even sure Mr. Anderson mentions it, but it's important because in the link reaction, that's where the pyruvates are turned into 2 acetyl groups, which are 2 carbons each, right? Which connect to a very important coenzyme A that escorts them into the Krebs cycle. So that is actually important. And another important thing about this is where some carbon dioxide is produced. You make a bit of carbon dioxide here. Remember, carbon dioxide can move through membranes with no problem. So that's a waste product, and that's going to be breathed out. And you're also making some very important NADHs here. We don't want to lose track of those because those do stay valuable. Those are going to be valuable when we talk about the end of this process. Are all the carbons accounted for? Take a look at this. We've got 2 3-carbon pyruvates Now we have two, we have the 4 carbons here and the other 2 carbons left with the carbon dioxides. So we are continuing to oxidize food molecules, to break bonds between carbons and hydrogens. And the electrons that are coming out of those are sometimes going into these electron carriers. 2 of them here, and then back out here, the glycolysis gave us some as well. So just kind of keep track of that. The next step is what we usually think of as step 2, which is that circular cycle, called the Krebs cycle. And a lot of people think of this as the second step. But remember that this link reaction happens in between, okay? The acetyl-CoA, that acetyl group, which is that 2-carbon group, is escorted into the first reactions of the Krebs cycle, and it gets changed along the way into intermediates until the same intermediate comes up to meet with the next acetyl-CoA. That's why it's cyclic. Memorizing the names of these molecules that it turns into is not that important, okay? What's more important is that you realize that the Krebs cycle is oxidizing the very last bits of those, of those food molecules, right? And taking those electrons and really reducing quite a lot of electron carriers. We make some of those, and we also reduce this other electron carrier that's also there. We also do make a little bit of ATP, not very much. We get 2 ATP, 2 more. And I'll also outline that in red so we keep track of it. There. And remember that these guys that I'm outlining in blue, these electron carriers that are all holding on to high energy electrons, whether they're here inside the matrix or whether they're hanging out here in the cytoplasm. They are the ones that are going to give us the biggest payoff in the end in terms of ATP synthesis. Okay, so let's go now to the third step. The third step, which is all important, and maybe probably the hardest one to understand. The third step in all of this is called the electron transport chain. Electron transport chain. And some people call it the ETC. So the electron transport chain, in terms of what it actually is, I remember this confusing me a lot when I was your age and learning this. The location and nature of the electron transport chain is right, bless you, is right in the inner membrane. There are these little series of very electronegative proteins. And I'm not going to draw them very accurately. I'm just going to point out that these little green circles that I'm drawing are, each one of them is an electron transport chain. Proteins that are embedded in the inner membrane. And there's not just one electron transport chain. There's thousands of them. Lots of them, sitting there waiting in the inner membrane for our reduced electron carriers to bring electrons to them. So there's a lot of electron transport proteins in there. And what do? They accept high energy electrons from NADH and FADH2. They accept them. And the second thing they do is they pump hydrogen ions. This up a little bit. Into the intermembrane space. Creating a electrochemical gradient. Remember the word gradient? Creating a gradient. Just give you a minute there. All right, so here's all these electron carriers. And in this state, in their reduced state, they're carrying high energy electrons. We learned about that. They're going to go over here and release those electrons and give them to these little green circles, which are the electron transport proteins. And what are electron transport proteins going to do? They're going to transport electrons across themselves like this. So the electrons are going this way, right? The electrons are going this way across, and all of them are doing that, right? So electrons hit them up here, and they all go in one direction this way, across, right? Wherever those, I mean, there's also not just one Krebs cycle here. There's lots of sets of enzymes running the Krebs cycle in here. So you have reduced electron carriers all around the matrix, right? So the electrons are delivered here and moved this way. They can only move one way. And at the same time the electrons are going this way, they are pumping hydrogens across into this space. And I'll make the hydrogens a nice color here. I'll make the hydrogens like pink. Here. So you're putting hydrogen ions in high concentration out here. And remember, a hydrogen ion is really just a proton, isn't it? Because a hydrogen atom is just a proton plus an electron. So what you're really doing is you're creating something that they often call a proton motive force. They also call it that. Where you're concentrating in this intermembrane space, you're concentrating a whole bunch of hydrogen ions. And remember, nature doesn't like that. Nature doesn't like things to be out of equilibrium. So what you're really doing is you're storing a bunch of potential energy in there. You're making a whole bunch of potential energy in the form of a bunch of molecules collected in that tiny little intermembrane space, just like pulling back on a spring. And the way that the energy is ultimately used is this. So in addition to these little electron transport proteins that I've drawn in green, there are these other special molecules that look like little rocket ships or keyholes maybe. And there's a lot of them as well. Oh, what a helpful noise. Okay, so these little pink things here are called ATP synthases. ATP synthase enzymes. And guess what they do? They synthesize ATP. So they're really well named, okay? And the really cool thing about them is that, remember, hydrogen's collecting over here. It doesn't like that. It wants to get back in. It wants to flow back down its gradient. And the only way back into the matrix is through the ATP synthases. That's the only way that the hydrogen ions can get back in. So anytime, let me draw this down here so that it makes a little bit more sense, because that's a bit busy. Okay. So let's say you have the inner membrane, and you have the outer membrane, and you have an ATP synthase here, okay? There's ATP synthase. And then you have hydrogen ions that are collected out here. And the reason that they are there is because the electron transport chain pushed them in there, right? So these guys here collected electrons, which flowed this way, and every time an electron flowed through this electron transport chain protein, it pumped some more hydrogen through, right? So ultimately, it's the electron transport chain that created this gradient, right? This is a gradient, a proton gradient. And I'm about to run out of time. Okay, in any case, every time this flows through here, ADP is phosphorylated into ATP. And that's